Okay, this is the second part of the review for the cell transport quiz. Okay, so number 72. When a cell releases a signal molecule into the environment and a number of cells in the immediate vicinity respond, this type of signaling is... Alright, so this is a definition question, and just to remind you, we kind of talked about three different categories of signaling, either when they're like close enough that the two cells are touching, or they're pretty near each other, or long distance. So we have direct contact, local signaling, and long distance. For the direct contact, we had the cell junctions, the gap junctions, or the plasmodesmata in plants, and the cell-to-cell recognition. For the local signaling, we have synaptic signaling between nerve cells, and then paracrine signaling, which is when a secreting cell acts on a nearby target cell. So that's definitely in the immediate vicinity, as opposed to hormone signaling, and which is endocrine cells, which is long distance. So immediate vicinity, but not direct contact. This is paracrine signaling. So the answer is C. Okay, number 79, chemical signal pathways. So this is kind of a definition type question. I'll give you a minute to read the responses. Okay, so it's not A because they occur in animals and plants. B, okay, they're not absent in bacteria. C, it's about the oh, barking. Hang on. All right, so the answer is D. Often involve the binding of signal molecules to a protein on the surface of a target cell. So that's just the definition of chemical signal pathways. In general, a signal transmitted via phosphorylation of a series of proteins brings a conformational change to each protein, requires binding of a hormone to a cytosol receptor, cannot occur in yeast because they lack protein phosphatases, requires phosphorylase activity, allows target cells to change their shape and therefore their activity. Right, so this is the kind of question that can really throw you because every one of them is so different, and if you don't know this vocabulary, it's really, really tricky. So the answer is A, okay? It brings a conformational change to each protein. So phosphorylation, if you need to review what that's all about, the phosphorylation cascade is on page 209. All right, and following that, they talk about the kinds of molecules involved here. And so they talk about, which brings us to the next one. So, all right, which of the following is widely used second messenger? Which of the following is widely used second messenger in signal transduction pathways? I think it's missing an as in there, okay? So A, calcium ions, B, cyclic AMP, and C, inositol triphosphate. So this is on page 210 and 211. So these are the three molecules that they talk about in signal transduction. All right, these transduction, again, is the cascades of molecular interactions relaying signals to target molecules in the cell. So it's like a big chain reaction. And so all three of these molecules are discussed in the chapter, and it goes through the process of their cascades. So the answer is E, A, B, and C. Okay, moving on to mitosis stuff. So we'll do a couple, or no, I guess this is the only one here that goes with with these choices of the phases. So in what phase do centrioles begin to move apart? Right, so if you're just holding on to the chromosomes and what the chromosomes are doing in each phase, that's definitely not enough. You have to know what all the parts of the significant parts of the cell are doing in each phase. So the answer to this is E, okay, prophase, so that's when the centrioles begin to move apart. All right, so before prophase, the centrosomes with centriole pairs are together at one end of the cell, and so they separate in prophase, and by prometaphase they're at the poles of the cell. Okay, number 94. A cell containing 92 chromatids at metaphase of mitosis would, at its completion, produce 2 nuclei containing how many chromosomes? So why are they asking you these things? So this is important, and this gets trickier when we're doing meiosis too, is to figure out how many chromosomes we start with, what is a chromatid, when has it been duplicated, what would we see in mitosis. Right, so when we're in mitosis, of course, we have already undergone replication, so each chromosome in mitosis is going to consist of each chromosome is going to consist of 2 sister chromatids, and so if we're talking at the completion of metaphase, when they're lined up along the plate, okay, we're going to produce 2 nuclei that are going to, oh, I'm sorry, I'm messing up this question because my dog is barking. The answer is D, 46, so half of 92, because the daughter nuclei are going to each have half of this, because during metaphase we're at doubled, and we're going to split into 2 cells. Right, so that means that the original cell in the G1 phase had 46. Number 95, if there are 20 centromeres in a cell at anaphase, how many chromosomes are there in each daughter cell following cytokinesis? So they want you to realize that at anaphase we've already separated, and the centromere is where? It's the region holding the 2 sister chromatids together with some proteins. So if it's already if the sister chromatids have already separated and we have 20 centromeres, then that means that following cytokinesis we're going to have 20 chromosomes. The formation of a cell plate is beginning across the middle of a cell, and nuclei are reforming at opposite ends of the cell. What kind of cell is this? So this is a definition type question about the cell plate. All right, so we're still in the chapter on mitosis. So a cell plate is only found in plant cells, and it's at the very end. Okay, so it is E, a plant cell undergoing cytokinesis. If mammalian cells receive a go-ahead signal at the hold on. Sorry about that. If mammalian cells receive a go-ahead signal at the G1 checkpoint, they will move directly into telophase, complete the cell cycle and divide, exit the cycle and switch to a non-dividing state, show a drop in MPF concentration, complete cytokinesis, and form new cell walls. So this is about understanding the cell cycle for mammalian cells. Well, if they get the go-ahead at G1, it means go on through and go through the rest of the cell cycle. So it's going to be B, complete the cell cycle and divide. Right. Cells that are in a non-dividing state are in which phase? So please make sure that you study the phases. So when are they not non-dividing state? Are A, the G0 phase. So here's a bunch of questions that are related to these molecules. So we expect you to be able to describe the function of these molecules in the control of cell division. So if you're confused about what these actually are, it's on page 229 and 230. So PDGF is platelet-derived growth factor. MPF is the maturation promoting factor, or the M phase promoting factor. Cyclin is cyclin, and then CDKs are cyclin-dependent kinases. Protein kinases are enzymes that activate or inactivate other proteins by phosphorylating them. Okay, and that comes from chapter 11, of course. So let's look at a few of these because I think it's important. Released by platelets at the vicinity of an injury. Well, it has platelets in the name, so yeah, that's A. All right. A protein synthesized at specific times during the cell cycle that associates with a kinase to form a catalytically active complex. Catalytically is difficult to say. A protein synthesized at specific times during the cell cycle that associates with a kinase to form a catalytically active complex. Well, what's that? Well, that is D. So that is cyclin. Okay? So cyclin is a major part of the cell cycle. All right. Here, how about this one? Nerve and muscle cells are in what phase most of the time? Okay. They're, of course, in the G0 phase. The shortest part of the cell cycle is what? It's the M phase, the mitosis, the mitotic phase. A mutation results in a cell that no longer produces a normal protein kinase for the M phase checkpoint. Which of the following would likely be the immediate result of this mutation? So this question wants you to understand when in the cycle the M phase checkpoint is. And it occurs between mitosis and cytokinesis. So you would have to know that whether or not it occurred before mitosis or after. So the answer is E. The cell would undergo normal mitosis but fail to enter the next G1 phase because it's occurring after mitosis. Density-dependent inhibition is explained by which of the following? I'll read these to you since they're kind of cut off. A. As cells become more numerous, they begin to squeeze against each other, restricting their size and ability to produce control factors. As cells become more numerous, the amount of required growth factors and nutrients per cell becomes insufficient to allow for cell growth. As cells become more numerous, the protein kinases they produce begin to compete with each other, such that the proteins produced by one cell essentially cancel those produced by its neighbor. As cells become more numerous, more and more of them enter the S phase of the cell cycle. As cells become more numerous, the level of waste products increase, eventually slowing down metabolism. So density-dependent inhibition, if you remember from, what page is that on? It's on page 232. And the answer here is B. As cells become more numerous, the amount of required growth factors and nutrients per cell becomes insufficient to allow for cell growth. so the cells stay at a particular concentration and level. Which of the following is or are true concerning cyclin-dependent kinase CDK? CDK is inactive or turned off in the presence of cyclin. CDK is present throughout the cell cycle. CDK is an enzyme that attaches phosphate groups to other proteins. So this is pretty much a definition type question of CDK. So CDK has a bunch of small facts to learn about. You should know that the activity of CDK rises and falls with changes in the concentration of cyclin. CDKs function by phosphorylating substrate proteins that affect steps in the cell cycle. Although they don't know what all the CDKs do, scientists have identified steps of the signaling pathway that involve CDKs. So the answer is E, as we talked about, that CDK is present throughout the cell cycle. It is not turned off in the presence of cyclin. And, that's the end, because the rest goes on to genetics in this section. So I hope this was helpful to help you review and look at some questions. And good luck on your exam.